Okay, it is audible. Is it audible? Okay. So with regard to this biotechnology part, so far we completed this up to what it is the BT cotton. And biotechnology is the production of nothing but what it is the biologicals, biopharmaceuticals. The biologicals, it is the food products, veterinary products, agricultural products. And biopharmaceutical, the drugs. By utilizing genetically modified organism, the plants, animals, the bacteria, the fungi, and the biotechnology having several applications in several fields. Diagnostics, the quick diagnosis, therapeutics for therapy, the drugs, and food processing like yogurt and cheese, and solid waste treatment, and bioremediation, that is reducing the effect of what it is the pollutants. So all it is the biotechnology application. And critical area of the research, the critical area of the research in this production of the best catalyst in the form of improved micro or purified enzyme. So in order to produce the product, we are providing optimal condition for the catalyst to act to produce the product. And third aspect it is the product produced, we have to extract it. So extraction of the product by using different downstream processing. That is one point. And in agriculture, inorganic agriculture, organic agriculture, and it is biofertilizer, that is inorganic and organic and genetically modified crops. In inorganic agriculture, that is agrochemicals they are using, pesticide, insecticide, fertilizer. And in case of organic agriculture, we are using varieties, living microbes to increase varieties, the fertility of the soil. And genetically modified crops. So, the non-conventional is genetically modified crops. And green revolution. In mid-1960, tremendous increasement, tripling of what is the food supply. What do you call it is the green revolution. Green revolution partly because of what it is improved, that is imported and it is improved varieties. Mainly because of what it is agrochemical, pesticide and fertilizer and better management practices. And other also contributed, it is better irrigation facilities and the other things which are also contributed for varieties, land reforms also contributed for green revolution. For developing countries like what it is India, the farmers unable to, that means put expenses on what it is the agrochemical. So it is highly expensive on the part of the farmer, especially in developing countries. And also it causing what it is the pollution. So is there any other method we can advise these farmers with the knowledge of what it is genetics and molecular biology? Yes. We can switch over from green revolution to gene revolution. Species specific barrier overcome. We can transfer the genes from unrelated organisms like bacteria into the plant is also possible by recombinant DNA technology. Why there was a great revolution in recombinant DNA technology? Because discovery of what it is restriction enzymes. So species specific barrier overcome. Now we can transfer the desirable gene even from the bacteria into what it is the plant. That is one point. And the organism, bacteria, plant or animal or fungi, whose genes which are manipulated by insertion of the foreign gene, what do you call it is genetically modified organism. And the genetically modified organism, GMO, genetically modified organism. Suppose if you take crop, GM crop, The crop is genetically modified. Genetically modified means its genetic makeup it is changed, manipulated, deliberate manipulation. What do you call it? Genetic engineering. So the manipulation of the genes in the organism results in what it is GMO, genetically modified organism. Apart from it is having the use in case of yield and resistance, it also contributing for what it is tolerance to abiotic stresses, the first point, temperature, drought, and salinity, the tolerance, the first one. Post harvest losses are tremendously be reduced. More shelf life period in case of variety the tomato, flavor sour tomato, bruise resistant tomato, delayed ripening the tomato, having more shelf life. And high nutritional quality and vitamin A rich rice, that is golden rice. And to resist abiotic and biotic stresses, that is basmati rice. And the GM crops increasing the efficiency in utilization of the nutrient, not in absorption of the nutrient. As efficiency as it got increased. Once efficiency it got increased, early depletion of nutrient from the soil is going to be what it is reduced. That is the advantage. Beside having advantages, they are genetically modified crop, tailor-made plants. The tailor-made plants especially suits to our specifications, like it is starches, fuels, pharmaceuticals. So what do we call it is genetically modified crops, that is tailor-made plants especially for certain purpose, for industries, the starches, fuels and pharmaceuticals. That is one point. In first one we covered insect resistant, that is Bacillus thuringiensis, certain strains of Bacillus thuringiensis, that means gram positive bacteria. It produces toxic proteins in certain phases of the growth. That is also key point. So Bacillus thuringiensis is not supposed to produce toxic protein at all stages, only certain stages of the growth. What do you call it is toxic protein. These toxic proteins which are insecticidal toxic protein. The insecticidal toxic protein do not harm to the bacteria. It is inactive. What do you call it is protoxins. So inactive toxins produced by the Bacillus thuringiensis Bt, what do you call it is protoxins. And this Bacillus thuringiensis spores are available in sachets. It made available in sachets in market, and these spores present in the sachet combined with what it is water and sprayed onto the fruit trees like brassica, cotton, tomato and potato. And whenever insect ingest it, in the gut of what it is larvae, the alkaline pH, the insecticidal protein, protoxin, solubilizes, crystal protein solubilizes and attach it and attach it to midgut epithelial cell, create pores, swelling and leads to lysis and finally death of the particular insect. Particular insect is going to be killed, that is caterpillar larvas. That is in abbreviation it is LCD, that is Lepidopterans, Coleopterans and Dipterans. In case of Lepidopterans, that is tobacco budworm, and it is armyworm. For this Coleopterans, beetles. For this flies and mosquitoes. So it acts on particular insect group. So it is narrow insecticidal application, narrow spectrum application. Okay. And it is the conventional method it is. And not, so what is the triggering environment in this alkaline environment is available? Triggering environment, solubilization of crystal protein into active toxic protein. Whatever the genes responsible for the production of the toxic protein, the genes are isolated from Bacillus thuringiensis and cloned into the plants like it is cotton, potato, and tomato to make it resistant for what it is insect. So, so pest resistant plants which are produced BT cotton, BT brinjal, BT tomato. And the choice of the gene depends upon the crop and target pest. Cry 1AB, corn borer, 2AB, 1AC, cotton bollworm. And this indication cry indicates what it is the proteins, crystal protein. The shape of the protein is the crystal. And this representation cry, the genes encoding for what it is the crystal protein. So it depends upon what it is, the target pest as well as what it is crop. To that extent, we've already covered in case of what it is Bacillus thuringiensis. Okay, up to it this is. Okay. And other it is the pest resistance. A nematode Myliodogain incognitia. Myliodogain incognitia. It is a nematode. And this nematode, the parasitic on plants, animals, and humans. It parasite is plants, animals, and humans. This Myliodogain incognitia, and this causes it affect roots of tobacco plant. So yield is going to be reduced. So Myliodogain incognitia, it is a parasitic on plants, animals as well as humans. As it affects the roots of what it is the tobacco plant, the yield is going to be what it is reduced. Okay. So in eukaryotes, there is a mechanism. There is a mechanism that is RNA interference. RNA interference. Otherwise, RNA quelling. Otherwise, RNA silencing. RNA interference, RNA quelling, and RNA what it is the silencing. Okay. So we know that RNA responsible for the production of what it is, mRNA responsible for the production of what it is, the protein. How gene is going to be expressed? By synthesizing what it is, the proper protein. So the nematode infect what it is, the host cell, makes the host cell to synthesize the protein required by them. So whatever the protein required for its activity, we are going to inhibit the production of what it is, the protein. So we have to inhibit the transcription or translation, we have to inhibit what it is, the translation. So when RNA is single stranded, it is going to make what it is, the protein undergoing translation. If RNA made double stranded, what? It prevents what it is, translation. So making RNA silent, that is nothing but RNA interference, RNA quelling, RNA silencing. So it's a defense mechanism, a nucleic acid resistance. So it is a defense mechanism. A nucleic acid imparts what it is resistance in many of what it is eukaryotes. So RNA interference, RNA quelling, RNA silencing, the different words. So parasitic on plants, animals as well as human. So which in case of roots of the tobacco infected by a myeloid, it is nematode, it is myeloidogyne incognitia. As roots are affected, as roots are damaged, there is no absorption of water and minerals. Ultimately, the plant is going to be what it is affected. So yield is going to be what it is reduced. So this myeloidogyne incognitia, the myeloidogyne incognitia, it infects and infest roots. It infect and infest varieties roots. It infect and infest varieties the roots. So first it is infect, it damages it. What do you call it is infest. So infecting and infestation, it is combat. You can combat it by a nucleic acid resistance. So RNA interference, that is RNA made into what it is, double stranded RNA. RNA made into double strand RNA. So prevent what it is, translation. Okay, it is. So RNA interference, what do you call it is? RNA made into double-stranded RNA, so prevent what it is, the translation. Particular protein not available for the myeloidogyne for what it is, the infestation. So how you are going to make it this RNA into what it is, double-stranded RNA. So making RNA into double-stranded RNA, it is nothing but RNA interference, RNA silencing, RNA killing. It is RNA interference is a type of nucleic acid resistance. Nucleic acid resistance. So here, how complementary RNA it is available? how complementary RNA is available by means of, so other RNA it should be made where it is available. So how other RNA is going to be made, it is available. Infection with the virus having where it is RNA genomes. That is one method. And we have to incorporate the genes into the host organism in such a manner that it should produce an RNA that is going to what it is making RNA into double strand RNA. You will see what is RNA interference, how RNA is going to become what it is, the double stranded what it is RNA. So it is mRNA 5'3" mRNA. If it undergoes translation, what is going to be formed? Particular protein is formed. Particular protein is formed. So this protein it makes the nematode or what it is infestation. Nematode infestation, it increases. So as required the particular proteins inside the host cell. So we have to prevent what it is translation, production of the protein, and protein it should not be available to what it is the nematode. So making the protein not available to what it is the particular parasite. We have to do that. So we have to prevent what it is translation. So to prevent translation of mRNA, it requires what it is other complementary RNA. Suppose it is 5'3". It become what it is double stranded RNA. So complementary RNA. How this complementary RNA is available? Suppose it is A, G, C, U, C, G, A, G, C, U. U, C, G, A, G, C, U, C, G, A. So we require what it is complementary RNA to make it double stranded RNA to prevent what it is translation. So source of complementary RNA. How the source of what it is complementary RNA. So once it made double stranded, it prevent what it is translation. And you come across a word in case of the molecular biology. If RNA becomes what it is double stranded, it prevents what it is translation. Even though transcription occurs, it is going to be what it is futile. That is going to be what it is waste. If DNA responsible for the production of what it is two RNAs, the two RNAs which are complementary, even though it is involved in the production of the two RNA, as the two RNA which are complementary, it becomes double stranded, prevent what it is translation. So entire transcription is going to be what it is futile. So here also translation prevented because of what it is double stranded what it is RNA. How the complementary RNA it is made available. And source of complementary RNA. The source of complementary RNA. That is infection by virus having RNA genomes. It is one thing. Infection by virus having RNA genomes. One. And using transposons, that is, what do you call it, is jumping genes replicated via RNA intermediate. It is the DNA. And okay, now you observe here. Within the DNA segment, the genes, the DNA, it is integrated into some other portion of it is the DNA. So what do you call it is transposons. So illegitimate integration of the DNA. So it is illegitimate integration of it is the DNA. That means the DNA of this, it is integrated at what it is some other portion. So it results in what it is some other characters. What do you call transposons? What do you call jumping genes? And here using transposons replicate via RNA intermediate. So DNA formed on what it is RNA. So RNA dependent DNA polymerase, that is reverse transcription. By reverse transcription, how many strands of the DNA found? Only one strand of the DNA found. If one strand of the DNA is formed, as nucleotides are readily available, it makes variety double stranded varieties the DNA. So, using transposon jumping genes replicate via RNA intermediate. In generally, transposons is what do you call jumping genes. So, DNA integrate at some other portion of the DNA, illegitimate transfer of the DNA within the DNA, illegitimate integration of DNA within the DNA, what do you call it is transposon, jumping genes. And it replicate via RNA intermediate, what do you call it is retransposons. It is generally it is transposon. Replicate via RNA intermediate, what do you call it is retransposons. So, transposons replicate via RNA intermediate, what they call retransposons. So it is RNA on which single stranded DNA is formed. The single stranded DNA is going to become what it is double stranded DNA. This double stranded DNA going to integrate it is the genome of host cell. And this is going to make what it is RNA. That is going to make what it is proteins. So what do you call it is retransposons. We are not having the mechanism of how it is going to produce. So it is RNA dependent DNA polymerase. So RNA dependent DNA polymerase. What do you call it is cDNA. What do you call complementary DNA? cDNA becomes what it is double-stranded DNA. Then it integrates into what it is its host genome, and it produces what it is RNA then protein. So it is one method. So transposons replicate via RNA intermediate. What do you call it is retransposons. That is one. Or infection by virus having RNA genomes. That is also what is one method. So for the ultimate aim is to make RNA to become what it is double stranded, that is going to prevent what it is translation. And by using agrobacterium, it is natural genetic engineer. Why you are calling it as a natural genetic engineer? Because it is a pathogen for what it is several several dicots. As it is pathogen for what it is several dicot. Ti plasmid, Ti plasmid, the tool, it is modified. The tDNA got modified. So this agrobacterium considered as what it is natural genetic engineer, it is going to cause the tumors, conversion of the normal cells into what it is the tumor cells. And by using agrobacterium, nematode specific genes introduced into host cells. In which manner? In such a way that it should produce sense RNA as well as antisense RNA. Then it becomes what it is double-stranded. So nematode specific genes are introduced into hosts in such a manner that produce sense and antisense RNA. If it sense and antisense RNA are formed, it becomes what it is double-stranded. In order to produce sense RNA, in order to produce antisense RNA, how many types of the genes are required? 2 genes, sense gene as well as what it is antisense gene. Sense gene produce what it is sense RNA. Antisense gene produce what it is antisense RNA. And both RNAs formed, it become double-stranded, prevent what it is translation. So nematode specific genes are introduced. So nematode specific genes are introduced in such a manner that it should produce what it is sense gene as well as what it is antisense gene. Suppose sense gene antisense gene is the DNA 3, 5, 5,3. It is antisense gene 3,5,5,3. So the sense gene is going to mean what it is. Sense RNA. Antigen gene is going to make what it is. Antigens RNA. Suppose in this sequence it is having okay. So in order to so 5,3 having the sequence it is AGC UAC G. So antigens RNA should be what it is U C G A U G C. So it produced the antigens RNA like this in reverse direction 3,5 UCG AUG C. So it forms what it is double stranded. So as it is RNA produced in which direction? 5,3 direction. Transcription in 5,3 direction. Translation in 5,3 direction. So mRNA produced in 5,3 direction. In reverse, it is going to be what it is the complementary. So it become what it is double stranded RNA. So AGC UAG. So the sequence of what it is RNA similar to that of which strand it is coding strand. So T. So here it is the DNA. It is T. Okay, A, C, G, A, T, G. So T, A, C, G, G, C, A, U, T, A, G, C. So it is template. So it is template, it is coding, which is sense RNA. In this antisense RNA, it is antisense gene. So it is CGU AGCU. In case of U, it is T. Here also it is T because it is the DNA. G C A T C G A. So guanine, cytosine, guanine, adenine, thymine means it is uracil, thymine it is adenine, cytosine, guanine, cytosine, adenine, thymine means what it is uracil. So it is the sense gene, it is antisense gene, sense RNA, antisense RNA, double stranded RNA. So no translation, no protein. So nematode can infect but do not infest because it is having RNAi mechanism. So the plant with RNAi mechanism, nematode can infect or infest, it can infect, but it do not what it is infest. Okay, clear idea with what it is the sense gene and antisense gene. the sense and okay, noted. Rest of members, noted it is sense gene and what it is antigens gene. Natural genetic engineer it is agrobacterium tumefaciens. It is pathogen for variety several dicot plants. Okay. So it is the sequence like this template and it is a coding. So mRNA sequences are similar to that of what it is coding strand. Coding strand in which direction? 5-3. 5-3. Template in case of it is 3-5. It is 3-5. Okay. So nematode specific genes are introduced in such a manner that it should produce sense RNA as well as antigens RNA. So sense gene and antigen gene produce sense RNA and antigens RNA respectively. It becomes complementary, double-stranded RNA. It prevents what it is translation. Okay. Tobacco with RNAi mechanism no myeloid can infect. True or false? Tobacco with RNAi mechanism, no myeloid can infect. It is false. Nematode do not know about RNAi in plant, can infect but do not infest because no proteins. Okay, now nematode do not know about RNA in the plant. It can infect, but do not infest. It cannot survive because no proteins.